Assalamualaikum warahmatullahi wabarakatuh.
Waalaikumsalam warahmatullahi wabarakatuh.
Kita bahasa Indonesia saja. Okay, good afternoon all. Welcome to the guest lecture today in the course of BOSA for our department and our grateful that you are willing to join us here in our guest lecture today. Today we have Prof. Fazira from University of Malaysia, Terengganu. Thank you, Prof, for being willing to join and share knowledge with us today. Prof. Fazira will give a lecture entitled Designing of Bioassay for Complete Clinical Studies in Botanical and Drug Development. But first, I will read Prof. Fazira CV. Prof. Fazira is a chemist at Institute of Marine Biotechnology, UMT. Prof. Fazira obtained her diploma in science from Institut Teknologi Mara Shah Alam Selangor, and then Bachelor of Science in Biotechnology from University of Leeds, UK, and then Master degree in Biochemical Engineering of University of Birmingham, UK, as well as her PhD in Chemical Engineering at the same university. Prof. Fazira's academic career began as a tutor at Bioprocess Engineering Department from UTM, but currently she is a professor at Marine Institute Biotechnology, UNT. That the brief CV of Prof. Fazira. And I will welcome to Prof. Fazira to give today's lecture. And Prof. Fazira, time is yours.
Bismillahirrahmanirrahim. Assalamualaikum warahmatullahi ta'ala wabarakatuh. And a very good afternoon to everybody. First of all, I would like to know the student. Can all the students introduce yourself? Anybody want to volunteer to introduce yourself first?
Yang ambil bioassay, tolong perkenalkan dirinya satu-satu. Nadila, silakan. Open mic.
Hello, Prof. Can you hear my voice?
Yes.
Yes. My name is Nadila. I'm a student in Magister Chemistry. I'm student Bioassay today.
What is your first degree? What was your first degree?
Bachelor of Education, Chemistry Education.
Chemistry Education, okay. So you're going to be a docent after graduation?
Yeah, inshallah.
Okay, thank you.
Dearly, please. Open mic.
Assalamualaikum warahmatullahi wabarakatuh. Mohon izin, Prof, saya izin memperkenalkan diri. Nama Dirli dari program studi Magister Kimia. Untuk S1 saya farmasis.
Okay. Very good. I can give different assignment based on different background. Okay, very good. Okay, next.
Next, Ulfa.
Assalamualaikum warahmatullahi wabarakatuh. My name is Ulfa. I'm from Magister of Chemistry. My bachelor same with Nadila. Jadi S1-nya sama kayak Nadila.
Chemistry education, ya?
Yeah.
Okay.
Student BOC only 3, but some of
Only
PhD
3.
students join here.
Okay. Okay. So I think I better start my presentation. Let me share my. Okay, can you see my presentation?
Yeah. Yes.
All right. Okay. Bismillahirrahmanirrahim. So saya diberi amanah oleh Pak Herman Shah untuk berkongsi tentang bioassays, ya. So I understand this class is for S2 and it carry 2 credits, okay. So before we start, I would like to share with you how I'm going to deliver the class, okay. So this is the course content. For today, I'm going to do bioassay 1, which is introduction to bioassay, the principles in bioassays, type of bioassays, and also the tools in bioassay. So today is not going to be very heavy, but next week for week 2 bioassay 2, we are going to do this together, meaning that we're going to design bioassay for a very specific study of maybe of your own choice. So I'm okay because I only have 3 students, so I can monitor each and every one of you closely. Because initially I thought there will be many students, so it's a bit difficult for me to manage the student, but only 3, then you are now in big trouble because I will follow you every single day. Okay. And then also next week, I'm going to show to you the bioassay for drug development. And fortunately, because at the moment, I involved in the development of a botanical drug for Malaysia, and I will share with you the real bioassay studies that we have been conducting for the past 6 years, and from the beginning, from in vivo, in vitro, in vivo, and inshallah next month is going to be clinical study. So I will share with you the insight into the whole process. Okay, and also in the week 3, I will do the case study. So since only 3 of you, so we'll do a very interesting case study. Maybe after second week, next week, I will give you an assignment for you to do. Then in the week 3, you will present to me your findings. Okay, so and that will carry the marks, yeah. Okay, so that's how it goes. I think if Pak Herman or the docent have any comments on this, please let me know, because I was told by Pak Herman Shah that I can have my own jurisdiction, on my own style to do the assessment. So, but if it is not suitable or need improvement by the faculty, please let me know. Okay. So, okay. So for today, we are going to look into introduction to bioassay. Yeah. Okay. A very simple one, because these days the information is just at the fingertip, so you can just always Google. I think if you are interested, I think you already know much more about bioassay than me, because you can always read online. So okay, but in the nutshell, bioassay as being defined by Oxford language, Oxford language dictionary, bioassay means measurement of the concentration or potency of a substance by its effect on living cells or tissues. Okay, with this definition, however, these days, the bioassay has been expanded. So there were the definition of bioassay has been expanded, and the living cells or tissue has been expanded to also the organs, the animals, and also human. Yeah, and also other organism. Any biological organism we can use to test our substance. Okay, and when it comes to a substance, what kind of substance that we use in bioassay? It could be chemical-based compound, maybe in the crude or in the pure form. It could be organic compound. It could be non-organic compound. Yeah. And for example, we have like here I put gallic acid, a very simple compound. And I'm sure by now everybody, all of you know what gallic acid is. Okay, very simple aromatic compound. Yeah. Okay, and then also it could also be a biological base, which is enzyme. It could be metabolites. It could be small peptides. It could be mRNA. You know, like today we have a vaccine that being distributed all around the world. It has been made with RNAs. Yeah. Okay. And then the compound can be in the form, it can be from natural resources, or it can be also synthesized chemically. Yeah. So usually the test substance or the test samples or active compound, some people call it, it could be meant to be developed into drugs, vaccine. It could be poison. It could be also toxin. It could be pesticide, cosmetics, cleaning products. It could be your body shampoo. Yeah? It could be antiseptics and so on. So that's the definition of bioassay. So is in the in the simple term, bioassay is any substance, anything that you want to test how it's going to affect in a living system. But when you talk about bioassay, it must have a proper measurement. It has to have a very highly standardized method. Yeah. Okay, so for example, when we talk about the substance, of course, it could be, in my example, I put gallic acid, but it doesn't have to be gallic acid. It could be the derivatives of gallic acid. It could be natural sources. It could be non-natural sources. Okay, when we talk about measurement, the measurement in bioassay, okay? Always when we want to do bioassay, we have always to know what will be the end point that we want to achieve. Because we can have many substances that we want to study, but the measurement that we want to do is depend on what we want to know about the substance that we are testing. Meaning that if we have gallic acid, what we want to know about this gallic acid in terms of when we put it in the biological system, in human body or in rats, or when we expose to the cells, things like that. Okay, so if we think, oh, we want to know whether or not gallic acid will be good or will have any effect on oxidative stress, on dyslipidemia, hepatostenosis, or could be anti-melanogenesis, anti-inflammatory. So we need to know the endpoint. We call it all the table, all the boxes here, we call it as an endpoint. This endpoint will determine the way we're going to measure our gallic acid, and also will determine what biological system that we have to use in order to measure the performance of this gallic acid. If you have a question while I'm delivering this lecture, please notify me, yeah? Okay. So next, yeah, okay. When we talk about living system or the biological system that we use to test our substance, okay, usually or most common we use these cells, okay. Can you see the cursor that I'm moving? Can you see the arrow?
Yes, sir.
All right, okay. So usually we will use this one, a cell-based assay for bioassay. But of course, depending on the effectiveness or the most suitable system that very close resemblance to human. Of course, people are changing the usage of the biological material. Okay, so before we go further, I would like to share with you also that actually bioassay is actually a method to know how the substance work in human body, because the ultimate goal when we do bioassay is for us to know how the substance is going to work in our human body, okay, usually, or if the substance is meant to be a product for animals, then we want to know how it affect the animals. So but for human, ethically we cannot use human to test for any toxin or any substances without having a series of bioassay using non-human first. Okay, that is by law, but ethically we cannot directly test any substance that we have directly to humans. So we have to do this bioassay first using non-human system. Okay, so the example we have here is cell culture system. This is the cell culture system is the system that I use during my PhD. Okay, and also we can use biopsy the organs or a piece of organ or piece of tissue from our body. For example, I show here this is the human skin that has been cut out and then being cultured in media. The pink one is the tissue culture media. So the cells are growing, the tissue is still alive. It has no soul, but the cells are alive. So we can do the testing as well. And also more recent one, we have this a 3D cell system that can be used for bioassay. The reason why people move into this 3D skin system, because the skin system will be closer resemblance to the human skin rather than just a monolayer culture of the cells. Yeah. Okay. So moving forward, after we finish our bioassay study with 3D or cell based, usually we will forward into the bigger organism. For example, the rat. Yeah. So rat is very common to be used for bioassays, and recently there are also a big movement in studying the whole biological system using zebrafish. Zebrafish are very interesting because the embryo is transparent. So any changes when we expose our substance into the embryo, any changes we can see simply under microscope. I used to have a group of students working on the bioassay using the embryo of zebrafish also. Okay, and also very interesting that zebrafish can be used to study the blood flow, the heart rate, the skeleton, and so on. So it's very interesting a living system for bioassay. And of course, ultimately, we have human as our living system to test our substance. But of course, before we can use human to test our substance, the substance usually already gone through all this lower living system of bioassays. Yeah. Okay. So moving forward. Okay. Now, a very interesting substance that has been newly developed, I think, for the past 2 years when we have COVID-19, and suddenly within 1 year there are many vaccine being produced, many different types of vaccine using different types of substance. Yeah. So for example, we have attenuated virus, inactivated virus, virus-like particles, protein vaccine, replicate viral vector, non-replicate viral vector, messenger RNA vaccine, DNA vaccine that been developed and being tested. Okay, now I'm going to ask you. Okay, I already asked you in the scenario of SARS-CoV-2 vaccine, the substrate are those vaccine. But what are the living system and what are the measurement that we use to measure the performance of the substance? Anybody wants to answer? Who we have here? Nadila? Do you want to answer? No? Anybody else? Who wants to volunteer to answer? What are the living system that we use for this vaccine bioassay?
Perhaps we use rat, ma'am?
Yeah, yes, rat.
Yeah, yes. Or mice and also monkey.
Yes. Yeah, probably. But I haven't read any report on the testing on monkey. But of course, you know, like today, the living system that directly being used to test SARS-CoV vaccine is actually us, the human, because actually to develop vaccine, it will take a very long time, maybe 10 years, because we have to undergo preclinical and clinical studies, clinical study level, phase 1, phase 2, phase 3, phase 4. But because SARS-CoV, COVID-19 is a pandemic, so United Nations and all the health regulatory authorities around the world, in every country around the world, they have to agree that this substance can be directly tested onto human. That is why we can get access to our vaccine very fast. But actually, the study is still ongoing, because if you are taking Sinovac, if you are we are taking Pfizer, Moderna, you are taking a different kind of Johnson and Johnson, the data, we are part of the living system in this big bioassay study. Okay. And my second question, what was the measurement that being what has been measured to indicate that the substrate is performing in this living system? What has been measured in our body? for this big worldwide bioassay study on SARS-CoV vaccine. Ulfa?
I think the antibody level on
Yes, correct. It's antibody levels. The immune reaction is correct. The antibody level. I would like to share. You know, last week I measure my antibody level, vaccine antibody level. My antibody level is just 30, I think 30 something, 30%. It's quite low because I took 2 complete doses of Sinovac. I don't know in Indonesia what are the common vaccine that you are using. Is it Sinovac or Pfizer?
Sinovac and AstraZeneca.
Ah, okay.
And with Moderna for booster.
Ah, okay. In Malaysia, Pfizer will be the booster for Sinovac. I have to take booster as well. I'm waiting for them to call me up. All right, okay, very good. Moving forward. So this is the principle in bioassays. Okay, it's very simple. Actually, bioassay is actually to compare potency of unknown substrate. Well, sometimes they say unknown, but usually it can also be a known substrate. But we must have standard. In bioassay, everything has to come with standard. Yeah, okay, and also the assessment will be including the measurement of errors. Yeah, and also the second principle of bioassay is standard and test sample should have same pharmacological effect and mode of action. Maknanya yang pertama, bila kita ada satu substance tu mestilah kita kaji bersama dengan satu substance lain yang telah kita kenali, yang telah maybe internationally recognized. The simple bioassay usually for example, if you want to measure phenolic compound, for example, or we want to measure the performance of certain organic compound like gallic acid. Gallic acid usually being used as a standard to measure alongside with the unknown substance or maybe known substance, but we just want to know the potency. Yeah, and also the another important things is that the samples, our substance or our test sample must have similar pharmacological effect with the standard. So if a gallic acid we use to measure phenolic content or to measure anti-inflammatory, for example, then we have to use if we want to measure, for example, if we want to measure the performance of X compound for maybe for diabetes, for example, for lowering blood glucose. So we must use a standard that already known worldwide or internationally known to have the lowering glucose effect. For example, we can use a compound, the active compound in a common drugs. For example, metformin being used clinically for lowering blood glucose. Then we can use metformin as our standard in doing bioassay for lowering blood glucose. Yeah. So I hope you understand that. That is why we have to be very careful when we design bioassay. We must know exactly what will be the best standard to be used alongside with our sample. That will determine how strong our data to be. Yeah. Because usually if you're talking about anti-inflammatory, maybe you want to use allopurinol is another substance for substance of being used for a clinical drug for anti-inflammatory. So those kind of things lah, yeah. Okay. Oh, what is this? Okay. So usually, as I mentioned, bioassay need to have standard. So when we plot graph for bioassay from our bioassay study, usually we must have standard. Okay, so usually, for example, in this case, I refer this graph from this paper. Yeah, differential cytotoxicity of Triphala and its phenolic substituent gallic acid on human prostate cancer cells and also normal cells. So, okay, so actually normal cell is our standard. Yeah, because we want to measure whether or not this Triphala has effect on prostate cancer and what happens if we expose to normal cancer. Okay, so usually, if you see here, we can see that when they are doing the testing of Triphala, they look at the viability. Viability meaning that when you expose the cells to this Triphala extract. So the first set of samples, you just put the cells, no Triphala inside. And then the second one, you put 10%. Then in this case, you put gallic acid, sorry. You put gallic acid at 10 microgram, and then you increase 20 microgram, 30, 40, 50, and 80. Then you see a direct impact, which is the viability. You count the cells. You maybe if you look into the microscope, then you will look, oh, the cells are dying. You can see the cells are floating. So you can measure that. You take it out using a very specific protocol, and then you can count how many cells that is viable and how many cells are dead when we expose the cell to gallic acid. So that is how. So also the second example I put here, the graph in the lower part, they test gallic acid to normal cells and also to prostate cancer cells. And they want to see whether or not when we increase the concentration of gallic acid, whether or not the viability of the cell will decrease or not. So in this study, they see that when there is no gallic acid in the cells, in the cell culture, there will be no death. It's 100% viable. And when you put 10%, the cells are still very much viable, no cell death. But when you increase the concentration to 20, 30, 40, 50, and then eventually 80, you can see a significant drops in cell viability. So this is the bioassay that we are looking for. There is a standard here and also the exposure, the measurement of the exposure using viability. Viability percentage is the measurement that we made in this particular study. Okay, we will see more example after this. Okay, so the types of bioassay. There are two types of bioassay, which is direct bioassay and also indirect bioassay. But usually, when we design our bioassay study, bioassay experiment, usually we will measure both directly, direct, and also indirect, because when we measure only one particular measurement, sometimes the data is not strong enough to support our study. Yeah. And also in our indirect study, usually we will have either quantile or quantitative data, but usually also both will be in our indirect study. And we using this, when we have this quantitative data, usually we have to measure very mathematically. There will be some mathematical modeling or mathematical exercise that we need to do, whereby when we plot the graph, we will have slopes, we will have all sorts of graph pattern, and we can measure the performance or the pattern of the graph to know as part of our measurement to know the performance of our substance. Yeah. Okay, so let me show you. Okay, the direct bioassay is actually, for example, we have here the structure. This is eupatorin compound. When we expose the eupatorin compound to the cells, we can see 10 micromolar of eupatorin will significantly reduce the number of viable cells. And when we increase more to 30%, we see a lot more cell being died by the exposure of eupatorin. And there was no cell death in the control because we didn't introduce any substance. Yeah. So when we plot the graph, when we count the cells, the viable cell against the dead cells, so usually we will have cell viability, and we can plot when at times at zero concentration, we will have 100% viable. And when we increase the concentration of eupatorin from 1 micromolar to 100 micromolar, we can see a significant drops in the cell viability. So this is a very direct measurement. Direct measurement is something that you can observe, you can see under microscope. So this is very interesting subject. Yeah. Okay. Indirectly, indirect bioassay. This is actually the photo is a direct bioassay, but this cell viability and the calculation on it, we can say almost going to go into indirect. Okay, so when we want to measure indirectly, usually we will look into what's next or what's beyond the eye observation. So usually, for example, if you are looking into the influence of this is gallic acid, for example, in inflammatory activity. So for example, here we make another measurement whereby we measure the presence of cytokines such as TNF alpha, NFKB, P65, and we use control beta actin as a control protein when we measure the cytokines that being produced in the cells when we expose to the gallic acid. Okay, so for example, in here we have the graph. This graph is actually also the indirect bioassay measurement. Yeah. So from here, we can actually plot the IC50 value and so on. Okay, next week I will teach you how to measure or to calculate this. Yeah. Okay. So next, okay. So in more to make you more understand on this bioassay things. Okay. This is actually when we expose, for example, gallic acid lah, yeah. When we expose gallic acid to cells that we put under the cell wall, 96 web plate, okay. So or 6 web plate or in our T flask here, okay. Then after a few hours, or usually we use 24 hours, 48 hours, and 72 hours. When we after we expose to a certain period of time, we will look at the morphology of the cell. This is we call it direct bioassay. So we can see in the beginning, most of the cells are viable, and then perhaps this is there is no exposure of gallic acid. This is a control, and then this is at certain concentration of gallic acid, you can see a lesser amount of cells, and this is also we can see a lot of cells, maybe at different concentration, and this is another concentration. So but when we do the bioassay, the morphological data alone is not enough. So okay, we can do another assay, which is we call it cell death. We can see the green, we expose the same cells, we add in ethidium bromide as propidium iodide or acridine orange. Acridine orange will stain dead cells, and then propidium iodide will stain the cell, the viable cell become green. So you can see the green is meaning the cell is alive. Yeah. Okay, and then you can see also the arrow showing that the cells are not healthy because there was the fragmentation of DNA when we look deeper using the microscope. Okay, and we are not satisfied yet with this observation because we cannot quantify. So now we have another equipment we can use, which is this is a flow cytometry analysis. We collect the cells, we do some treatment, and then we, azan in my home is very loud. Okay, so bear with me. So in the normal cells, we can see that cells are saturated in just one corner, and when the cells are being exposed to gallic acid, so you can see the distribution. You can see a different distribution at different exposure. Yeah, and also when they do another staining with MDA, so they can see significant difference. And from the data, from the flow cytometry data, they can actually, I'm trying to hide this one. Okay, so when we have this treatment, okay, so you can see this is, we can actually do a quantitative analysis whereby when we expose these cells to a certain condition, you can see 100% apoptosis. Apoptosis meaning that cells are going to die. Yeah, the condition where cell is going to die. So when they put gallic acid in the dark condition cells, so you can see there were reduced amount of apoptosis, and some cells are able to move to proliferate. Yeah, you can see apoptosis, maybe this one, other early apoptosis, late apoptosis, and also necrosis. And also when they put a C and laser, so you can see also significant apoptotic death. And then when they measure gallic acid together with the laser exposure, they can see they could minimize the apoptosis as well. So this is an example of the data that we can get when we do bioassay. Okay. but it's all depend on what equipment that we have in our lab. Okay, sometimes we cannot be so excited to do you know testing, but we don't have the equipment in our university, for example. So we have to modify our research. Okay, quantile versus qualitative bioassay. What does it mean? Okay, quantile meaning that when we do the measurement, we usually will have a specific measurement. We will know the effective dose at the 50%. Then also we can know what happened, what is the effective dose for 90%, 80%, something like that. And we can use software to measure the LC50, LD50 values, and these are the example that we can get. I will use this journal next week for our exercise. Yeah. Okay. So for qualitative data, qualitative bioassay, usually we measure the physical effect. For example, in this zebrafish system, this is the work done by my student previously. So this is a normal embryo of the hatching embryo of the zebrafish. It's very beautiful. It's very clear. This is just using a normal microscope. Yeah. Normal light microscope, and this is the embryo when we expose the embryo to a safe concentration of our substance, and this is the embryo when we put higher concentration of the substrate. So we can see there are some teratogenic effect, yeah, whereby the yolk sac being enlarged, and the embryo, the hatching has edema. So so we know that the substance has influence in growth, and also when the concentration is too toxic, the embryo, the eggs is not hatching at all. It stops at certain stages in cell replication. So yeah, this is another types of bioassay that we can do. We can analyze the morphological effect of our living organism that we are using. Okay, it's very interesting. Okay, we're also looking at what is the meaning of slope ratio versus parallel line model when we do the quantitative assay. Of course, this is very much into pharmacological study, perhaps not that crucial in S2 for natural product, yeah. But I just share with you anyway. If we are talking about the slope ratios, okay, slope ratio means that when we expose different concentration of substance to our living organism or living we call it a model, yeah, bioassay model. Okay, we will see a pattern of performance, yeah, pattern of response. So it could be this way, it could be this A, could be B, could be C, or could be D, or could be any pattern, yeah, depending on the data that we generated. And of course, when the slope are different from one into another, we know that the substance are very much different. But if we use the same substance with the different concentration, we should expect to get similar pattern, similar slope ratio, only the distance will be different. For example, the A and the C, the slope A and slope C, we could say this is of the same substance but different concentration. And this A and C, we call this as a parallel line model. And And then BACD, we call it as a slope ratio. I mean, of course, it has a different slope ratio. Yeah. So when we of course, by having this slope ratio and also parallel line model, we can make prediction of the fate of the substance that we are using. Okay, I will not go into detail yet on this, but usually by you having this slope ratio or whatever a pattern that we have, we usually good pattern, good we call it a good and logical pattern will give us a quite significant indicator that the substance is working. Okay, like that lah. Okay, so for example, like this one at 0.1 microgram drug dose, we can see it has a 50% effect on the drug response. Then if we give 10 microgram, so we expect that this is the maximum tolerated dose, for example. This is very much into pharmaceutical calculation. Yeah. Okay, next, tools in bioassay. I don't know how much time do I have.
Our lecture is about 100 minutes.
100 minutes?
Yes.
I thought it's
credit is 50, but it's up to you, Prof.
Oh, I only prepare for 45 minutes.
Okay, it doesn't matter.
Is it okay? Okay, never mind. We will have a question and answer after this. Okay. I think another important things that we need to know is the tools in bioassay, because sometimes when we I know that eventually the student will do the experimental work, and of course when you read journals, when you read other publication, oh, they do this way, they do this way, but sometimes we want to do but we cannot do because maybe certain equipment or certain things are not there. So we have to modify our research a little bit. Okay, so the tools in bioassay will depend on type of substrate, type of living system, and also the type of measurement that we want to see. We call it what are the endpoint that we want to measure. Okay, if our substrate maybe not that much, but we also need to know if the substance is pure, if you want the substance in the crude form or things like that. So we have to have it will determine also the bioassay that we're going to do, because sometimes certain substance cannot be dissolved in water, cannot be dissolved in certain solvent, and if we use the wrong solvent, and it will be the solvent itself will kill the living system, things like that. So we have to be very careful in selecting the tools that we're going to use. And also we have to look into the physical tools. Yeah. For example, if we decide to use cell-based bioassay, so we must have a cell culture lab and the related equipment. When we want to open cell culture lab, of course, it's not just we buy one biosafety cabinet and we can start. No, it's a lot of other things that we have to prepare first. I remember in year 2000, when I came back from my PhD, I was given a lab to set up a cell culture lab. So I have to do a lot of work in order to gather all the related equipment, the must-have equipment first, so that I can start my cell culture work. Okay, so we also need a biochemistry analyzer, for example, the microscopic devices. Biochemistry analyzer is for us to measure what's happening in the cell culture, things like that. And microscopic usually we use to measure the morphological aspect of the our test samples. Yeah. Okay. So another important things that we need to have is the statistical data analysis software, which help us a lot in presenting we call it statistically accepted data. Yeah. Okay. So I give you an example of this cell culture laboratory. This is very beautiful one. Do we have cell culture lab in Unsri in Mipa?
Cell culture lab in chemistry we don't have.
Cell culture lab you don't have in Mipa or in Sriwijaya? Maybe other faculty have.
Maybe other faculty.
Okay. Never mind. When student going for research work, maybe you can send your student to different lab in campus.
Yes.
Yeah, because if to develop a tissue culture lab, cell culture lab is not cheap. So and it's also take long time. So and university will not invest for the new lab if the similar lab is already exist somewhere in the university. So just send student. Okay. Okay, so of course we have to have this lah in the simplified version, but we need to have all those equipment. Usually this lab is very clean. We cannot bring any samples like any raw samples inside this lab because it offer contamination. Okay, so usually we want to come into this lab, we only can bring our already finished samples like the extract in the vial that has been sterile, and then come and do the work over here. Okay, and not many people can mingle around in this lab. Usually depending on the size of the lab, so student maybe only 1 or 2 students at a time. Okay, because tissue culture lab is very easy to get contaminated with microbes or with fungus. So we have to get rid of all those contaminant away from our lab. In fact, if your lab is next to microbe lab or in fungal lab, no, it's also not good. You have to be a little bit isolated. Okay, or else it's easily contaminated. Okay, so these are the list of equipment that usually needed depending on what measurement that we are going to measure. Yeah? Okay, for example, of course, HbA1c analyzer is for diabetic studies, hematology analyzer for to assess blood samples, ELISA reader for any antibody link bioassay, chemistry analyzer if you want to test ALP, AT, or all those body chemicals. Centrifuge is a must in tissue culture lab. ESR analyzer, hemoglobin meter, blood gas analyzer, coagulator. Microscope is wajib ada. Automatic tissue processor optional. This one also optional. Cryostat microtome, because this is when you have animals to study, because we do biopsy, we cut the tissue with this microtome and cryostat microtome for preserve the tissue. Incubator is a must. Hemodialysis machine not important, it depends on what you want to study. The fridge and freezer is a must. The ionized water is the wajib. urine flow meter, not necessary depending on the study, maybe human study this one. Clean bench, it has to be clean bench is for you to work with the samples preparation, but biosafety cabinet level 2 minimum is biosafety cabinet level 2 you must have for cell culture study. Ultrasonic nebulizer, this is also important. Homogenizer depending on the study. This one water rocker also depend. Water distiller wajib ada, water bath wajib ada, autoclave wajib ada. So this is equipment besar. Equipment kecil-kecil lagi, wah, so many. Okay. So another important things when we want to work with biological system, for example, the cell culture techniques, we have to master the technique first in handling cells. During my PhD study, of course, during my S2, I already master myself with the cell culture techniques. And of course, when I do S3, it's very easy for me because I already understand how the cell culture work. Of course, if you are working with animals or with zebrafish or other living system, you have to really understand those systems first before we can conduct any experimental work. Yeah. Okay, so the good things when you do your mastery S2 is for you to get mastery in techniques, yeah, in a specific specialized techniques. Yeah, please do that. So okay, apart from that, we know already what we need to have, what kind of bioassay that we can use. So there will be time that we need to design our experiment, bioassay experimental design. Before we sit and design our experiment, we must do this one thing. One thing that you have to do is to think or to decide the ending of your story before you start. What this mean? Decide the ending of your story before you start. Meaning that if you have a substance, you have to know what you want to know about this substance. Only then we can design our bioassay experiment. For example, as I already shared this slide before, for the gallic acid. Now, okay, if we want to know the anti-inflammatory activity of gallic acid, then we design the bioassay specifically for anti-inflammatory. If you want to do anti-HBV, anti-acne, we want to use gallic acid for cosmetic, for skin whitening, for skin rejuvenating, then there will be a separate bioassay to be done. Okay. If you want to know whether gallic acid is good for antimicrobial, then there will be a different study. We have to use microbes system for our assay. Okay, so in the nutshell, if we are studying herbal drugs or any component or compound in the herbs, so the first thing that we have to do is to do the extraction process. It doesn't matter what kind of extraction solvent that you want to use, but usually if you intend to use your substance as a botanical drug or as to be used to be consumed, ya, untuk dimakan, the best is for you to use water extraction. You use is aqueous extract, yeah, or you can go for what we call medical grade that is very expensive, or food grade alcohol for extraction. The rest, like other solvents like acetone, or other things, those solvents are only for chemical assay but cannot be used as a medium to extract the compound and then use that compound as a drug for oral consumption. If you have to do other things that to ensure the purity of the substance. Yeah. Okay. So usually when we have a different kind of extract using different solvent, because I know when you are in natural product, it is very common for us to do successive extraction, right? So you put same samples in different solvent and then you extract the samples. That is common for us to know what are the compounds in that particular herbs. Okay, it's just generally we want to extract everything from the herbs or from the plant. Okay, so but if we want to know how active, what are the activity of this extract, then we have to design the bioassay. But this bioassay has to be specific to what you want, because if you want to know the potential of a drug for diabetic, but you design the bioassay for anti-inflammatory, it will not work. It's not to say wrong, but it is not correct. Okay, salah lah. Okay. That's why we have to know our bioassay very well. Okay, so if the bioassay is not active to what we want to see, so we have to discard and then we use the active one. And usually when we found the crude extract to be active, and then the next step, what we'll do commonly, we do the fraction. We call it, we get active fraction, maybe fraction 1, fraction 2, fraction 3. This method can be, this fraction can be obtained by chromatographic method. And then with these separate fractions, we do another bioassay and see which one will be active. And the active one will be proceed into purification process. But it also depends. If your purification, if you really want it to be pure to become a candidate active compound, to become an active compound for your drug candidate, then so be it, you purify it. But if you want it to become just a botanical drug or just to become what we call it in Indonesia is a product terstandar, yeah. Bukan phytopharma, or you just want it to become a phytopharmaca, then you can just use active fraction. That is sufficient. Okay. And then with these purified samples, you have a few isolate, for example, so then you test again. The same bioassay, you test again which one are active. So the active one, then you can proceed into either dual chemical structure determination or do another sort of assay. But to tell you the truth, from herbs to this active purified compound, oh, it will take a lot of time, as well as the amount will become very much less and less and less and less. Okay. So alternatively, what we can do is when we have extract, okay, we can do analysis using more high-end equipment like LC-MS. Then we can read the profile when we put our extract into this LC-MS. Then we can have a very busy peaks with a lot of compound inside. Then we can decide, oh, which compound is more prominent. Then we can target that prominent compound to be used as our lead compound. Yeah. So from there we can do this scoring using we call it webometric analysis. Not webometric. Okay. Chemometric analysis. This chemometric analysis, then we can see which compound is active and which one are not active. So, but we have to conduct also a specific bioassay. So from here, we can make another analysis. Then we can find our active metabolites and that complement with active samples. Then from there, we can do further analysis as well using bioassays. Yeah. Okay. So what else that we have to do? Okay. Right. Okay. Important stages in bioassay experimental design. So now we talk quite a bit on the theory part of the bioassay. Now we want to design our own bioassay for our experimental work. So for example, here we have our plant. Maybe you will be given a plant, and then you must do the extraction and make a fractionation, and then you get the crude samples. For example, this one is endographolite enriched extraction. It's not pure, but it has a high concentration of endographolite. Do you know what is endographolite? This is endographolite is a active compound that is extracted from Endographis paniculata. In Indonesia, they call it sambiloto. It's very bitter.
Yeah.
Okay.
Yes, sambiloto.
It's very common. Okay, so usually, so far there's a lot of study being done for endographolite, because this endographolite, this sambiloto, has been used as traditional medicine for the past 2,000 years. It is used by Chinese for Chinese medicine. It's being used in India for Ayurvedic medicine. It is being used in Europe. In Italy, they use endographolite extract, the Endographis paniculata extract for fever. And in Australia also they use they already approve endographolite extract for fever as well. And in Thailand now, they use endographolite to be prescribed together for COVID-19 patient that the COVID-19 in the early stage, stage 1, stage 2, 3. Okay, so there's a lot of potential of endographolite, and you can see here the already known information regarding endographolite is anti-cancer, anti-diabetic, hepatoprotective, analgesic, anti-HIV, anti-malaria, antibacterial, antiviral, anti-inflammatory, and so on. But apa lagi yang nak dicari? Okay, apa lagi kan kita nak tengok? Oh, mungkin kita kata okay. Maybe for the novelty of your research, we want to look into cardiovascular disease maybe, because it's not being studied yet. Yeah. So if you want to do cardiovascular disease, which bioassay to use? Which living system that you can get access to? Can you do cell-based assay? Can you do animal study? Can you do human study? Not yet. Can you do zebrafish system, or what are other system? Can you use microbes? Do you have a microbial lab in the faculty? So these are the things that you also need to consider. Okay, what is available in your university or potential laboratories around you? So this is also important. And also you need to ask your supervisor, how much money your supervisor want to spend for your project. Okay. And also where you want to publish, because this will determine how intensive your study is, and how important how you need to design your experiment. It has to be your design has to be correct, has to be internationally accepted. Okay. So that only then we can publish in a very high impact factor journals. And which study level that you are. If you are doing S1, maybe the study just simple. At the S2 is a bit more complicated, but when you do PhD or even more complicated bioassay that you have to do, or even a lot more things like that. Yeah. So yeah, these are the things that we need to consider before we excitedly, want to start our experimental work. Okay, so for example, if you are looking into this cardiovascular related effect of endographolite, for example, in using a cell-based model. So I browsed through just now 1 hour ago, 1.5 hours ago maybe. How is there any paper? Is there any published paper that study cardiovascular disease using a cellular model? So we found yes, there are so many technical paper already published with a lot of with a different cell-based model to study cardiovascular disease. So you can actually read these journals and see whether this methodology suitable for you or not. Yeah. Okay, should you think, oh, I want to do zebra fish system as a model of my living system for endographolite, for example. So, okay, I also look into a zebrafish cardiovascular disease model. So kita tak payah fikir-fikir nak design sendiri unless we really determine maybe at a PhD level we maybe create our own model system. But usually for the ease of our study, so we follow a standard method, much easier, yeah. Okay, especially at the S2, so just follow what others have done. Okay, so there is a lot of study that use zebrafish system as a disease model for cardiovascular disease. Then we can take 1 or 2 papers and read the protocol, what is the methodology being used, then we can follow. Okay, and then last one, maybe we say, I want to try animal model. Maybe maybe in Unsri you have animal house. A lot of people are studying substance using animal model, so we can create cardiovascular disease animal model to study cardiovascular related disease. But bear in mind, when we talk about cardiovascular disease, it's a very big issue. It's a very big thing. And when we want to do our research at master level, we have to narrow down and to scope our work into a very limited aspect. Yeah, we cannot do everything under the sun. So we just select what will be the most suitable for us to do within a period of 1 year, for example. Yeah. Okay. Or perhaps say, okay lah, since endographolite has been used, a lot of study has been done in animal model. I want to use human model. Okay. So directly nak buat clinical study, boleh ke tak? Boleh tak agak-agaknya? Jawapannya tak boleh. Okay. Human model has to be a clinical study. But unfortunately for endographolite, there is not yet human clinical study being done using endographolite specifically for cardiovascular disease. Yang lain-lain untuk demam, untuk COVID-19 pun masih in silico work. Ada banyak report lah, tapi in human model, before we design human model study, we have to complete our our preclinical study first, okay, using different bioassays as I mentioned just now lah. So with that, I think I already finished my presentation. So saya bukalah if anybody want to ask any question. Okay, before we continue next week with designing bioassay for specific study and bioassay for drug development. Okay, any question?
Siapa yang mau bertanya silakan langsung.
Boleh saja yang S3 pun boleh tanya.
Boleh, siapapun boleh ya. Siapapun
Yeah.
yang mendengarkan silakan untuk bertanya. Silakan raise hand. Boleh disampaikan dalam bahasa Indonesia ya, Pro?
Boleh, saya insyaAllah faham. Tak faham tolong translatekan.
Silakan. There is no question.
No question. Wah, everybody understand.
I want to share something
Permisi,
about
Bu.
is
Tadi ada siapa yang nak bertanya tadi?
Ulfa, Bu, mau nanya, Bu.
Oke. Ulfa, silakan Ulfa.
Jadi kalau ini pakai bahasa Indonesia ya, Bu?
Iya.
Silakan. Mudah-mudahan Prof paham.
Faham, insyaAllah.
Nah, jadi kalau misalkan penelitian pakai hewan atau animal, apakah ada perbedaan kalau kita pakai misalkan itu kan ada mencit, tikus, kelinci, atau tadi ada zebrafish. Nah, apakah kalau misalkan kita mau uji antioksidan, apakah ada perbedaan hasil kayak gitu kalau kita pakai mencit sama tikus sama kelinci sama zebrafish kayak gitu? Apakah kalau pakai mencit hasil antioksidannya itu sedang, kalau pakai tikus besar kayak gitu?
Okay. Dia depend on this very di sini memang menjurus kepada pharmaceutical kan. Mincit tu kecil kan. Tikus tu besar. Ke yang mana yang lebih besar?
Mencit more small.
Mincit tu kecil kan? Okay.
Yes.
Usually bila kecil, luas permukaan perisi padunya besar, and then the effect saya tak ingat dia punya calculation, tapi efeknya dia seelok-eloknya haiwan tu lagi besar, lagi mirip kepada human system. Okay, sebenarnya apa-apa yang kita buat bioassay ni sebenarnya dia ultimate goal adalah untuk kita test kepada human. And then bila kita study, kalau dalam pharmaceutical regulation for drug development, kita hanya memerlukan sama ada rodent, it doesn't matter is either you guna mincit atau you guna rat, but it has to be in a rodent family. One substance it needs to be done in two types of rodent. Two types of rodent, that mean you ada satu mincit, ada satu rat boleh. Ataupun satu lagi in rabbit. Rabbit also same rodent kan, ataupun guinea pig. And then another study you have to do is in primate or dia panggil non-rodent system. So there are two system that you need to do. But if you're talking about zebrafish, certain assay will be considered, but for safety assay, no. Okay, kalau pharmaceutical, rodent and non-rodent is a must test system untuk drugs to be developed for human use. Okay. Tapi biasanya kalau kita nak untuk structure function, kita panggil, kita nak faham apa sebenarnya yang berlaku dalam tubuh ataupun dalam badan bila kita masukkan satu substance. Contohnya kalau kita masukkan endografolite ke dalam zebrafish, kita nak tengok adakah aliran darahnya terhenti ataupun makin laju ataupun sebagainya. Jadi kita boleh tengok zebrafish system tu, sebabnya zebrafish tu dia sistemnya transparent. Jadi kita boleh nampak darah mengalir very clear. Memang kita nampak darah mengalir. So kita boleh capture the image there. But that is only for pharmacodynamic data. Maknanya kita nak tahu apa yang berlaku when we put our substance into the body, what it does to the body. Tapi tak memerlukan untuk dia menggunakan rodent and this non-rodent. Non-rodent, and you have to remember the non-rodent untuk pharmaceutical adalah usually adalah beagle dogs, anjing yang memang khusus untuk dijadikan animal study lah. Dia panggil beagle dog. Okay. The research to use this beagle dog is very expensive. In Malaysian ringgit is already like 2 million for a simple 6 days, 60 days or 90 days 6 month safety study, something like that. So very expensive. Even not many laboratories around the world yang boleh offer this kind of study. Jadi macam tu, tapi kita di universitinya selalunya masih di level rodent lah ya. And then study kita pun is not GLP, not in a good laboratory practice. So we can use the data for education purposes. But for immediate or for regulatory data untuk kita submit untuk kelulusan BPOM kalau di Indonesia kan, itu tak boleh lah, tidak diterima. Okay. Because they have a different requirement macam tu. But no problem, if we want to study for the mechanism, it's okay. Macam teratogenicity kan, macam kita guna tikus untuk tengok kesan substance kepada anak yang dilahirkan, things like that. So itu boleh digunakan mincit ataupun dah sekarang regulatory masih belum terima if you use zebrafish, but the movement is ongoing lah untuk orang saintis push the authority macam FDA, macam di kita di dunia ni memang kalau pharmaceutical dia ada regulatory body dia. Indonesia lain, Malaysia lain, tapi similarity-nya tetap ada. Dia orang memang asasnya tetap sama macam tu. Ada lagi soalan?
Okay. Ada yang lagi?
Saya Prof.
Saya tu siapa?
Nadila.
Okay.
Nadila, so I have a question based on bioassay measurement. We have indirect and direct, right? Which more effective between direct and indirect, or every single sample got different treatment?
Depend on what we want to see. But usually we have to use both direct and indirect, because if you have one sample, one substance, then you expose the substance to the cells, for example, yeah, and then you see the morphological changes, and then you want to report this as your master. Do you think it's enough? Are you very confident that this is the only data that already can support your study, or you need a few more studies to be done, few more data, to support your study? But usually there will be a few study that we need to conduct, and to qualify your substance to be accepted as having this kind of these therapeutic properties or whatever activities lah, Kok kalau you S1 mungkin dosen hanya suruh okey buat satu saja, tengok morfologi, nanti seorang lagi tengok cell death, macam tu, viability. Tapi kalau at S2 cannot, it's not enough. You have to do a lot more.
It's not enough.
Yeah, and then the data has to be lebih what you call it, has to have confidence. Kalau kita kan bila kita buat penyelidikan, mestilah kita mesti confident kan, kita nak confident dengan data kita. Kalau kata saya dah tengok morfologinya dia macam ada efek, takkan itu saja, mesti ada data-data lain to support yang bersifat kuantitatif, because when we do scientific study, quantitative data is more important. Sebab morfologi kualitatif data ni macam sometimes orang boleh query, orang boleh ragu, orang boleh ragui, jadi menyusahkan kita lah.
Okay, bro. Thank you.
Okay. Nadila di mana? Bunyi belakang tu macam di kafe.
Yes, right. In the cafe now.
Saya pun rasa seperti berada di kafe. Very nice.
Another question, please. We have ada yang
I want to ask Berlina Sari
Yes.
Sayati.
Silakan Ibu.
Yeah. Ma'am, I want to ask, I'm a doctor, study doctor. So I want to
doctor
ask,
meaning a PhD
doctoral
or
program.
Doctoral. Okay.
Yes.
S3.
Yeah, I want you to give me an example, the range or limitation, or how far that the research for the doctoral program.
Okay.
If I want to research an extract or one medicinal plant.
It depend on the objective and the scope of your study. For example, in PhD, usually we will measure the breadth and the depth, meaning that lebarnya ilmu itu dengan kedalamannya. Macam bila ada sesuatu discovery yang kita buat, mungkin daripada extract ini kita tahu ini bagus untuk maybe untuk adalah effect on certain mechanism yang you nak tengok, and then you have to go detail. Why? After you see, you can see the glucose being reduced, and the next question will be how. You have to answer how, because when we answer the how, meaning that we study the mechanism, how it works in the body, because we will understand the diabetes mechanism, and we must know how our substance affect the pathway of this diabetic mechanism. That will be the depth of the study. The scope is depend on what are the objective that you want, but usually if you have just one big objective, and then usually in PhD depend on the school lah, the school of thoughts. Ada yang usually kalau my student, saya hanya letak 3 saja supaya di awal-awal masa bila buat proposal tu mereka rasa wah senangnya 3. Tapi 3 itu tidak semudah itu. But the scope, we have to go very detail on each objective. Yang paling penting the depth. But in PhD, of course, you have to read a lot, and you kalau boleh cuba keluar daripada kepompong yang jangan jadi satu tempurung gitu. You have to be very broad. Kena berfikir di luar, kadang-kadang apa yang you dapat tu, what will be the application outside. Orang mungkin satu yang orang tak di luar jangkaan, kita boleh fikir macam-macam. Tak ada masalah dalam tidak berdosa kalau kita berfikir banyak-banyak. Asal jangan berfikir pasal zat Tuhan, sudah.
Okay, ma'am, thank you for your
Alright.
explanation.
Okay. Is there any question? I think there is no more question, Prof.
Yeah, you want to ask question.
I just want to share because I'm a natural product background. We also isolate pure compound, but according to your designing bioassay, we assay from the extract, and then the active extract, we further separation until we have a pure compound. Then and then we test the say again, and actually
similar to the diagram that I show you, yeah?
Yes.
Okay.
But
That's common for natural product research.
But actually, the compound, the pure compound we have isolate, usually not give the activity that we have wanted. Sometime that we isolate the isolation to isolate the pure compound is not easy. Yeah. Sometimes we can only isolate for the much more compound. For minor compound, sometimes we cannot isolate.
Yeah, understood.
It's the challenge, and we have a problem about this. And how about your experience?
Okay. It depends. I'm not from a pharmacy background, so I can do a much more interesting method. Okay. But for those who are from the line of pharmacy and natural product that is moving in the direction of pharmacy, of course, you always looking for crude and then pure active fractions, and then try to purify. And then when you found that when you purify, you get a very small amount, and then it's not active, then you start doing the modification, right? So you because you have done a lot, so the next thing to do is, maybe we modify and put things here and there, make a derivative, maybe it's more active. And then of course to do the isolation, to do purification is very expensive and also very time consuming. Why don't we do synthetic? That's where pharmacy coming in, because you cannot get access to the raw material, and you do a lot of series process, and you only got a very small amount. Not active. You active, you have to modify things like that. Okay, but another school of thoughts, okay, pemikiran berbeza-beza, ya. Okay, approach berbeza, tapi ultimate-nya adalah kita nak menghasilkan drugs ataupun produk yang bagus untuk human consumption, ya. So another things that the new movement, ya, bukan saya saja, tapi now worldwide, people or scientists or big pharmaceutical company are looking into multiple compound, no longer a single compound. Okay, the reason being, the logic behind this is because human body is a complex system. And if you only introduce one specific compound to target this one pathway, it will not be sufficient, because in order for you to rectify the disease or the problem in your body, it has to do a lot of, multiple very complex network that you have to address. So if you have a multiple compound, this multiple compound will do the work here and there. Satu pergi kaki, satu pergi tangan. So it works in a holistic manner. They call it a synergistic effect. So that is why these days even big pharmaceutical company is already looking into multiple compound, and not in a pure form, no longer in the pure form. As long as you can standardize, like you have a few phytopharmaca now in Indonesia that is already being clinically proven, but it is a combination of multiple phytochemicals. Not to say you add kemudian, bukan phytochemical 1, 2, 3 you tambah kemudian, but it's already in the plant, but you just use mungkin active fractionnya ataupun it's a standardized extract against certain phytochemicals because they believe it is more lebih aman ya kepada tubuh, and then and it works much better macam tu. Jadi saya tak surprise bila you akan menghadapi masalah ini, because big pharmaceutical company telah melaluinya, I think 50 tahun yang lalu, 100 tahun yang lalu. That is why pharmaceutical company semuanya end up with synthetic drugs, because for you untuk tebang pokok, untuk cari pokok quinine yang asalnya dari Indonesia, mana you nak dapat? Kalau you duduk di Poland, kalau you duduk di Rusia, because you tak boleh simply access, bukan you tidak lagi menjajah di Indonesia, then you cannot get the pokok tu daripada Indonesia, isn't it? Because that's why you have to create synthetics. But for us who are living in the tropics yang kaya dengan biodiversity, I think it is worth for us, our nation di Nusantara ni, we work differently from the farmer. But saya rasa idea ni mungkin akan mengambil masa mungkin lagi 50 tahun. Tapi seandainya, bukan seandainya, jika big pharmaceutical company telah pun making steps into this, takkan kita masih lagi nak ikut step dia yang lama. You know, because sebenarnya patutnya kita perlu lebih ke hadapan daripada mereka. Kita tak boleh tunggu, oh dia orang buat, dia orang guna polyherbs, baru kita nak invest into that. Mungkin masa itu dia orang produk-produk di Indonesia yang polyherbs ni mungkin datang daripada Pfizer, mungkin datang daripada Nestle is having also nutraceutical company, you know. At the end of the day, kita ni banyak ilmunya di Indonesia ni, di Malaysia pun, tapi orang lain yang take benefit. I think saya sangat emotional in this issue, because saya rasa kita yang perlu berubah dahulu, and then orang kita yang kita nak bagi sihat orang kita, bukan orang di Eropah. Kita ada cara kita, cuma kita hanya perlu put an effort untuk bagi saya kita mesti menghormati dan meletakkan herbal medicine kepada suatu tahap yang tinggi sesuai dengan the function. Cuma sekarang ni kita, sebab kita tak ada ilmu, kita tak tahu kalau jamu yang dijual, jamu gendong ke jamu apa tu, kita tak tahu dia punya effectiveness. Walaupun orang minum mungkin sudah 50 tahun, 100 tahun tak ada masalah, still fit, still okay. But without scientific data, kita tak boleh convince the younger generation because today generation, they are learned people, they read, they need to be convinced with evidence. So we need to look into this more rather than kita go to isolate and at the end of the day tak jadi, you know. Jadi ke situ saja, berhentinya di situ, gitu. Dan pokok lain pula sampai bila? Sampai pencen, sampai dah habis berhenti kerja pun tak ada satu produk boleh dikeluarkan sebab then I say buat tak jadi, repeat the same thing. Mungkin kita boleh start fikirkan untuk keluar daripada root itu. Macam itulah. Because saya dulu when saya ni backgroundnya biopharmaceutical. I studied monoclonal antibody production and when I came back to Malaysia, saya fikir sebenarnya because the technology is expensive and I don't think it's suitable for many people in Malaysia. I rasa there must be another way. Satu lagi yang saya belajar, when I want to induce production of monoclonal antibody tu, sebenarnya the ingredient that we put into the medium untuk grow the cell tu masih daripada phytochemicals. So it means that if we can eat phytochemicals and then our body can induce a lot more, produce a lot more. So I think that is more lebih benar, lebih betul bagi pandangan saya. That's why saya move into herbal medicine because I think it's more logical. Gitu. So itu pandangan saya lah.
Yeah. Okay, Prof. Thank you. I think it's fourth. It's time is up. Maybe there is no more question. Thank you for Prof. Fazila for lecture today.
Cuma saya nak tanya, my lecture notes to be shared with the student. I think maybe afterwards you can share my WhatsApp number to the student. Okay, to all students, can you take my number now?
Of course.
Okay, it's 6019 770 6882 6019770 6882. Okay, nanti simpan ni dan WhatsApp saya, nanti saya buat group. Saya akan buat group dan saya boleh share my slides to the student. I think I'm okay for S3 to join in maybe in our discussion also. I'm happy to help.
Thank you, Prof. And maybe we are join again in next Saturday.
Week? Yeah.
Okay. It's time is up. Okay. Thank you, Prof. Good afternoon. Assalamualaikum warahmatullahi wabarakatuh.
Waalaikumsalam
warahmatullahi wabarakatuh.
Jumpa lagi insya-Allah minggu depan.
Thank you, Prof.